Bueno, buenas tardes a todas y todos. Pues es un placer aquí hoy de tener a la doctora Laura Villanueva aquí. Em, de hecho, el seminario yo creo que sí entendí va a ser en inglés porque pues por el idioma universal de la ciencia, pero pues, está perfectamente bien. Yo de hecho también tengo mi semblanza que preparé está en inglés, pero voy a ver si la medio en Spanglish la logro hacer. Pues Laura es un research leader bueno, es por, no sé, el Royal Netherlands Institute for Sea Research, que se ubica en Texel, que es una isla pequeña cerca de la costa en los Países Bajos. Ella hizo su doctorado en ecología microbiana en la Universidad de Barcelona. Después fue un postdoc en el Departamento de Microbiología de la Universidad de Massachusetts. Siguió un segundo postdoc en el Center for Systems Biology en la Universidad de Harvard, en los Estados Unidos, y en 2009 empezó como tenure track, como científico en el Departamento de Biogeoquímica Orgánica Marina, en el mismo lugar donde está ahorita, y ahorita está como Senior Research Scientist en el Departamento de Microbiología Marina. Bueno, uno de sus intereses principales es la geomicrobiología molecular y lo que significa, para los que no conocen el término, que es de buscar entender a nivel molecular las actividades de ecosistemas en el presente y en el pasado. Bueno, es un placer tenerte aquí, Laura, presentando en nuestro seminario. Y pues antes de que te dé la palabra, nada más, pues la gente que nos sigue en el canal de YouTube puede hacer sus preguntas en el chat y nosotros los vamos a leer aquí y vamos a contestarlos aquí. Y las personas presentes en la sala de Zoom eh, pueden hacer como indicar que quieren hacer una pregunta con la manita virtual que tienen en el Zoom o poner un comentario en el chat que quieren hablar y nos les vamos a dar la palabra. Pues muchas gracias a todos por conectarse y Laura, pues adelante, estamos muy curious a ver qué nos...
Muchas gracias, Cristian, por la invitación. Es un verdadero placer estar aquí hoy y presentaros un poco lo que he hecho en los últimos años. Toda la investigación es gracias a muchas personas que trabajan en mi grupo y me debo a ellos porque realmente esto es un trabajo de equipo y no es de una persona sola. Así que voy a intentar acknowledge them durante la presentación. Me veo incapaz de hacer la presentación en español, de verdad que lo siento, pero no lo he hecho en muchos años y realmente preparo las presentaciones a little bit on the fly y me veo incapaz, pero por supuesto hacer las preguntas en español y si hay alguna cosa que no se entiende, por favor, hacérmelo saber. And with this, I'd like to start. Today I want to tell you a little bit about membrane spanning lipids, what they are and what do we use them for, their evolutionary implications, and a little bit of how we approach it. And Christian said, currently in the Royal Netherlands Institute for Sea Research, in the Department of Marine Microbiology and Biogeochemistry. And one of my core businesses and something that I'm very passionate about is microbial lipids. And as Christian said, since 2009, I'm a stage here in this small little island in the Netherlands. And what we aim in my group is to discover new microorganisms, especially microorganisms that are found in the marine environment, check if they have metabolisms that are out of the ordinary, but always with this thing in mind that we are looking for lipids, very special lipids in these microbes, lipids that we can use as biomarkers, that means as trackers of the presence of these microorganisms, both in the present and in the past. And how do we do this? We do this by a combination approach. To be able to analyze the lipids, we use organic biogeochemistry methods. But of course, we couple this with genomics and molecular evolution of how these lipids have been synthesized over the course of evolution and what can they tell us about the first organisms on Earth. And for doing so, we take really cool samples from the bottom of the ocean with our research vessels. And in the last, let's say, 5 to 8 years at the NEOS, I've been establishing a marine anaerobic microbiology lab in which we go out there in the field, we take samples and we isolate cool microbes in anoxic conditions and also under high pressure. And well, this probably most of you know, of course, our view of the tree of life has changed dramatically with the application of genomic methods. And this is the tree of life based on the publication of Laura Hook et al. In 2016. And that already showed you here labeled in red how little we know about the microbial life that it's out there, because only the ones that have been labeled in this tree with a red dot are those that they can be actually cultivated, and the rest we just know based on their genomes. And that is, of course, very interesting. But the tree of life that I really like is the tree of life based on microbes and their lipids. And lipids can be very diverse as well. Obviously, not as diverse as genomes, as the DNA, but still very useful. And in some cases, they can be associated to specific microbial groups. And many people sometimes they really ask me, like, well, I mean, you're interested in lipids, but everything can be done with genomics. So why do you really study lipids? And I force myself to put this slide very often in when I'm teaching and also in presentations to convey the idea of why lipid biomarkers are so cool. And we use lipid biomarkers I've been using them lately to answer evolutionary questions of how microbial life appear on Earth. Another particular characteristic of lipids, specifically of the membrane lipids that they are found in the membrane of microbes, is that when they are found in their intact form, they still have the polar head groups attached to it. But when a cell dies, these polar head groups fall immediately in the order of minutes or hours. And this is very useful because, of course, if we detect something like this in the environment, we know that this is associated with living cells. And this is not trivial because when you go to the environment and you get DNA, that DNA can be associated to living cells, but also to dead cells. Another thing, as I said, as some lipids have been found to be quite specific of certain groups. For example, there's this very special membrane spanning lipid that is called crenarchiol, and it has 4 cyclopentane rings and 1 cyclohexane ring. And this one has been only found in Archaea. But I think one of the things that is remarkably useful of studying lipids, microbial lipids, is that they can be preserved in the sedimentary record very well, much more than DNA. When you think about, of course, mRNA or ribosomal RNA, it can be preserved in the sediment in the matters of hours or days. DNA, if you are lucky, you can look at kilo years if the sediments have been very well preserved, what we call ancient DNA. But lipids, actually, they can be preserved millions of years. And due to this characteristic that lipids can be preserved in such high old-time scales, lipids can be used as paleoproxy. That means that they can be used as indicator of past conditions on Earth. And one of the things that I find very cool in the last years is this application of looking at membrane lipids in the perspective of evolution. And when you look at this tree of life, very simplified, of course, even like maybe not so updated. When you think about bacteria, eukaryotes, and archaea, there's something quite remarkable about the membrane lipids. And that is that when you look at the membrane lipids of bacteria and eukaryotes, they have the same structure. They have fatty acids linked with ester bonds to glycerol 3-phosphate, and that is exactly the same in eukaryotes. While archaea, actually, it has something completely different. We have isoprenoids linked through ether bonds to glycerol 1-phosphate. This different stereochemistry is what we call lipid divide, because there's a really big differentiation between the members of archaea and the members of bacteria / eukaryotes. But that, of course, brings to something else as well, is that within archaea, we do have a certain diversity. Archaea, they can have membranes that are composed of diethers. That means that we have this structure over here with 2 ether bonds. and they form what we call a bilayer. That's very similar to the phospholipid membranes. But Archaea, they are always known to have also these membrane spanning lipids, so monolayers that of lipids that they are spanning across the membrane. And these ones, they are called tetraethers because they have 4 ether bonds. And as I was saying, one of these controversial things about the acquisition of membrane lipids in the course of evolution is the lipid divide. Because at some point we assume that the last universal common ancestor had either one or the other type of membrane, or maybe both, and it had to diversify into the 2 kinds of membranes. But we don't really know how the membrane of LUCA, of the last universal common ancestor, was. Another thing that is quite controversial is that if we believe the latest evidences that point to Archaea as the host to generate an eukaryotic cell in eukaryogenesis, if Archaea, they have this kind of membranes, and eventually eukaryotes, they have the other type of membranes, you have to imagine that there has to be some sort of transition also between an Archaea-like membrane to an eukaryotal or bacterial-like membrane. So there's this kind of division of the membranes in the 2 types until there's a specialization. But that, of course, it has to go through certain stage, both this stage of the last universal common ancestor and the transition and the origin of eukaryotic cell. It has to go through what I call the most likely explanation to have a membrane that it has both bacterial-like membrane lipids and Archaea-like membrane lipids, or also heterochiral membrane. And this mixed membrane or heterochiral membrane, it was always believed to be impossible because they were saying, well, it's very unstable to have such a membrane. You cannot have a membrane composed with both types of lipids. That membrane is not stable, so it's not existing. But that was refuted not so many years ago, I think a couple of years ago, with this paper of PNAS of Caforio et al., in which they engineer an E. coli to synthesize Archaea lipids as well, up to 30% of the membrane, and that membrane was stable, not very happy, but quite stable. So that means that actually you can have a stable mixed Archaea bacterial membrane. It's possible. Nevertheless, we have never found any microorganism, neither contemporary or evidences from the past, that indicates that this transition, this mixed membrane transition, ever happened during the course of evolution. Another thing that it's also important to take into account is that this differentiation in the bacteria and archaeal membranes, it's quite like that. The rules never really mix up, but there's one exception, and that exception is that actually there are certain bacteria that they do also form these membrane spanning lipids, and when you look at them, they do resemble so much to these membrane lipids that I was just showing you before, the membrane spanning lipids of Archaea, or tetraethers, we call them GDGTs, but they are not really the same, because as they are bacterial, they don't have the glycol 1-phosphate stereochemistry, but rather they have the glycol 3-phosphate stereochemistry. These bacterial membrane spanning lipids, they can have tetraesters or tetraethers or mixed ethers and esters or lipids. But we do have certain representatives that they make these membrane spanning lipids. Among them, some acidobacteria groups, they make these membrane spanning lipids. Some members of the Thermotocae, Thermonaerobacter, and Clostridium, and Butyribibrio from Firmicutes. But that's it. We have not really found any more culture representatives that make them. And we still don't know what would be the physiological reason why they do have these membrane spanning lipids. And also we don't know if they are in any way evolutionary related with the membrane spanning lipids of Archaea. One of the things that I was saying that it's quite important for us is that lipid biomarkers, they preserve very well in the sedimentary record. And because of that, they can be very good indicators of the past. And we have been using this for quite some years. to actually reconstruct temperature. And this might be shocking if you don't know, if you've never heard about it, but we actually use lipids from Archaea that they have been preserved in the sedimentary record to actually reconstruct the temperature at which these Archaea were living in the water column of the sea. And Archaea, it's a group of Archaea that they are found ubiquitous everywhere, but in the ocean they are found in the shallow waters. They can be very abundant. They are chemolithoautotrophs. They fix bicarbonate. They oxidize ammonia. And at some point, it was determined that they make these membrane spanning lipids, as many Archaea do. But they make these, what we call GDTs, with 0 cyclopentane rings up to 4 cyclopentane rings. And they can also make Trenarcheo, which is this very special lipid with 4 cyclopentane rings and 1 cyclohexane ring. And when they look at the distribution of the Archaea lipids in sediments, they saw that the amount of rings, the different kind of distribution, it was correlated with temperature, so that you can actually reconstruct the temperature at which these Archaea were living back in the moment in which their lipids were imprinted with this signal. So we are very much focused on still using these lipids for paleotemperature reconstruction, but of course we are very much interested as well to know if there are other Archaea that they could be living in the water column of the sea or even in the sediments, that they could potentially be making these same lipids and that they could contribute to the signal in the sediment. And that would be bad for our paleotemperature reconstruction, but still we want to know. And for doing so, we actually want to know much more of which kind of lipids Archaea make, both for evolutionary purposes, but also we want to know, without having to culture them, which kind of lipids they make to account for potential signal in the sediment. As I was saying, there's also these bacterial membrane spanning lipids that in some cases they can also have 4 ether bonds. And these membrane spanning lipids of bacteria, they also have been found to be quite affected by the temperature. And these ones, they are found in soils everywhere and also in freshwater systems. And they are also, their distribution is correlated with temperature. And of course, due to their usefulness for reconstruct temperature, we want to know who's synthesizing them, because if we know which kind of microorganism is synthesizing them, we can account for maybe if they have preference for certain nutrient or certain part of the year, or yeah, anything. The way that we do this experimentally is that we have the problem that unless we culture a microorganism, we cannot really determine for sure which kind of lipids they make, because we cannot really extract their lipids and look at them under our instruments. And to be able to compensate for that, and sorry for the cursor because I just realized that this came from another slide. We actually target, when we cannot culture those microorganisms, we target the ability of those microorganisms to make the compound of interest. And for doing so, we target their lipid biosynthetic pathways. What we do is, of course, we try to identify potential candidate genes with bioinformatic approaches to see if there's any motif or any activity that we would like to predict that is involved into making that compound, for example, a methylation or a radical mechanism. And what we do when we have target a specific candidate gene is that, of course, we confirm the activity of this gene by either doing heterologous gene expression or gene knockouts. And when we know for sure that that is actually the gene that we're looking for, we screen the presence of the gene in other genomes of interest to infer which kind of microorganisms they can have the capacity to make this lipid. And eventually, if we manage to get a culture, we can confirm it, obviously, by lipid analysis. And when we are looking at the lipid biosynthetic pathway of membrane spanning lipids, we do actually have quite some knowledge, especially when you are looking at the membrane lipids of Archaea, these GDGTs, these membrane spanning lipids. We know that you really need to get some isoprenoid building blocks. So we know very well how you get the isoprenoids with the mevalonate pathway. You need to get the stereochemistry of Archaea. So glycerol 1-phosphate, we know that it's formed by glycerol 1-phosphate dehydrogenase. And the two key steps here is to make the ether bond between these isoprenoids and the glycerol 1-phosphate. And this is mediated by two prenyl transferases, geranyl, the glycerol synthase, sorry, GGP synthase, and a DGGP synthase that mediates the second ether bond. And after that, there's a series of steps for which sometimes we don't know anything. For example, we speculate that to get the membrane spanning lipid, you need the condensation of two diethers, but this is a carbon-carbon bond that has never really, it's a reaction that has never really been described, so we don't know which kind of enzyme that could be, and has never really been actually confirmed which one could be. There's, for example, other enzymes that have been recently tested, and we know, for example, that the enzymes needed to mediate the formation of cyclopentane rings are radical SAM proteins, GRS, but we actually don't know anything of how a cyclohexane ring, in the case of the Carrachio, is formed. So there's still many unknowns here in blue, and many things that we already know, at least for the GDGTs of Archaea. So when we talk about unknown membrane spanning lipids, when you talk about GDGTs from Archaea with tetraethers, these are the reactions that we would aim to know better, the ones in blue. When you're talking about these branch GDGTs, these membrane spanning lipids bound in bacteria, we basically don't know anything. We don't know how the ether bonds are formed, if they are there. We don't know how these building blocks would be joined, because you know for sure that they are not isoprenoids, that they are rather fatty acids. So we assume that there's a fatty acid coupling. And we also have cyclopentane rings in those, and also methylations, but we also don't know how those are formed. To determine the biological source of these membrane spanning lipids, either the Archaea ones or the bacterial ones, we have two approaches. Either we have a model organism that we know that it synthesizes the membrane spanning lipid, that we really want to know more about their biosynthesis. And that's, of course, the most ideal situation, that you know a model microorganism, that you can identify candidate genes. And how we do this is that we know, in some cases, we don't know anything about the pathway, but we, for example, know which environmental conditions, either temperature or inhibitors, the membrane spanning lipids, and that's what we do basically. Apply certain conditions in the model organism, do transcriptomics, do proteomics, and see which kind of candidate genes could be doing upregulation or downregulation. Another approach we have is actually go to the environment and say, okay, I know that in certain sample, I have a lot of the lipids of interest. So I'm going to get this sample. I'm going to do lipidomics. I'm going to do also methyl from this sample. And I'm going to try to correlate the presence of certain organism or certain lipid biosynthetic pathway, and also the presence of that lipid to infer who is actually making the lipid that I'm really interested on. Sorry about that. I'm going back to the previous slide. Okay, let me see what we can do about this. Yes. Sorry about the potential problems of this. Okay, yeah, that should work now. Okay, so yes. Well, that was nice that I got a break in talking in one slide, but let's go back to this. So I was just saying that we have Thermonarobacter ethanolicus as a model microorganism, one of the approaches that we do. And Thermonarobacter ethanolicus, this makes these membrane spanning lipids, what we call branch GGTs, but in this case, not the ones that are formed with tetraethers because they don't have the ability to make ether bonds. And what we know is that the Thermonarobacter ethanolicus makes these membrane spanning lipids, and the building blocks are isodiabolic acid, which is this resolved hydrolysis product of this membrane spanning lipid. So what we did actually, and this was work of Melvin Tsiliakos, a postdoc in my lab, is that he was looking for which kind of growth conditions, as I was saying, which kind of conditions could actually tease around the formation of isodiabolic acid as building block of this membrane spanning lipid. And what he saw actually, because this was unknown, is that just by simply growing Thermonarobacter ethanolicus, isodiabolic acid here in orange increase during the stationary phase to have a maximum amount in the stationary phase, and the formation of isodiabolic acid in this way was also coming together with a decrease of iso C15 fatty acid, which we already suspected just by simple math, because isodiabolic acid has C30 carbons, that the building block could be iso C15, but here we had a more substrate product relationship over the course of the growth phase of Thermonarobacter ethanolicus. We did this at two different temperatures, optimal and suboptimal temperature, and we could see that this was consistent. So basically, we had further evidences that the precursor of this isodiabolic acid, the building component of these branch GTGTs in bacteria, it was actually iso C15. And as a follow-up, Melvin Tsiliakos and another postdoc in my lab, Diana Sagonero, wanted to further prove that how the synthesis of isodiabolic acid was happening in Thermonarobacter ethanolicus. And what we actually did is to give the culture of Thermonarobacter labeled leucine, which is a precursor of tetradecanoic acid. And that what we were expecting is that actually this fully labeled leucine would get incorporated into label iso C15 and eventually into label isodiabolic acid. And that's what we actually saw, that in a time course, here you could see here in orange, the label iso C15, that it was increasing over time. And this increase of iso C15 was also coming together with an increase of isodiabolic acid label over time. So we had a further proof that iso C15 was the building block of isodiabolic acid. And now that we knew that that was the case, what we wanted to do is, okay, we have an environment, a grow condition that is around the formation of isodiabolic acid, the building block of these branch GGTs in bacteria. So Diana did some transcriptomics and also some proteomics based on contrasting conditions to actually see which kind of functions could be upregulated. And we did also some limitation for certain enzymes that we know that they might be involved in this process, which is the coupling of two C15s, iso C15, to get an isodiabolic acid, to be able to target potential isodiabolic synthesis. We have actually come to a reasonable list of isodiabolic acid syntheses, just between 10 or 12. And now we are currently busy because we have synthesized all these constructs to be expressed in E. coli, to be able to see E. coli that can actually synthesize isoC15 if we can come out with formation of isodiabolic acid that could confirm that those are the isodiabolic acid syntheses. And that, of course, it's of great interest because when we have actually confirmed the candidate gene, these isodiabolic acid syntheses, when we look already at the databases, already these potential ones are pointing up to many different kinds of organisms as potential sources of branch GGTs in the environment. And that is not a surprise for us, because when we were using these branch GGTs of bacteria for reconstruction of temperature or soil input, we could see that they were much influenced by many biological sources. So this is going to be really great for us when we can confirm which kind of microorganisms they have this potential, because then we can really constrain very well how we can use these biomarkers to reconstruct soil input and also temperature. And another thing that, as I was saying, another approach that we use quite a lot, and it works quite well, is that when we don't have a model organism that we can culture in the lab, a model organism that we know that synthesizes our lipids of interest, or in this case, membrane spanning lipids, what we go is go out there to the environment and get a lot of information about environmental lipids and the genomes, the producers that are out there, to be able to couple this information and to narrow down the potential producers. I have a particular interest of looking for Archaea as membrane spanning lipids producers in anoxic and sulfidic marine systems, just because they turn out that, of course, those systems are extremely cool for other reasons, and we can find really new metabolisms that we didn't know about. We go to the Black Sea. The Black Sea is it's an euxinic basin. Here you have labeled the euxinic basins that are throughout the world in blue, like the Baltic Sea or the Namibia or Cariaco Basin. And the Black Sea, it's a very special system. It's what we call the largest euxinic marine basin. That means that it's has a deep water that is anoxic. It has also high concentration of sulfide. It's permanently stratified, doesn't mix up. It has a redoxcline. That means that it has a region in which oxygen and sulfide coexist, but it's almost no oxygen. It's around 100 or 150 meters depth. It can have sulfide concentrations up to 400 micromolar in the bottom waters, and it's very stable. So we know that it's a great source of microorganisms, and especially of Archaea. And that's why we went there. And here you have a little bit of the profile of how the Black Sea looks like. You have oxygen up to more or less 70 meters. You have what we call the redoxcline or suboxic waters. And then around 105 or 110, we hit the euxinic waters in which oxygen is not detectable anymore and sulfide builds up. And here you have more or less the sediments. Actually, if you can recover water from the water column, you can recover sediments. The sediments are really very well preserved, highly amounts of organic matters, and very laminated. So really nice to see. And what we did in several campaigns, but this one touches upon a campaign in 2013, is that we went out there, we got waters recovered from 15 depths across the water column of the Black Sea. We got samples for DNA extraction, for RNA extraction, for lipids. For lipids, we did high resolution accurate mass spectrometry combined with lipidomic analysis. And for DNA, we target who was there based on 16S amplicon sequencing, but also we did methygenomes with Illumina to be able to reconstruct metagenomic derived genomes. For these, of course, when you go to an environmental sample, you have many lipids that you have never really encountered before. And the approach we had until very recently is that we were analyzing which kind of lipids were there based on what we could identify as something that has been described before. It can be phospholipids, Archaea lipids, SQDGs, vethain, you name it. But there's actually, when you go out there and when you look through high-resolution accurate mass spectrometry approaches, you get a lot of components, what we call features, that they are mass spectra, that we know that they correspond to lipids, but we cannot really assign them to a specific lipid just yet. But we can still use this data to reconstruct potential clustering of these features with certain depths. And this is work of Nicole Bale and Sudin, also in my group. And they actually analyzed the water column of the Black Sea from 50 to 2000 meters depth. And they saw certain clustering of the lipids, of the features, based on the different areas in the Black Sea, which in a way it kind of makes sense because the lipids are synthesized by specific microbes that they like to be in the different areas of the Black Sea. But what I found extremely useful and powerful of this method is that we can use the lipidomic data, these features, to perform molecular networking of this lipidome, for example, of the Black Sea. And there are certain features that they have network connections with other lipids that we know that we have already identified in the past. For example, here you have a feature that we know that it's a sphingolipid and that it clusters with something else. So there's certain relationship that allows us to identify unknown compounds, these features that we had never really encountered before. And we can further use this and correlate it with the presence of lipid biosynthetic genes, but also with the presence of certain microbes to kind of associate the features with the sources. So when we went to the for the first time to the water column of the Black Sea, actually we were with a white book because we had never really, since the 80s, nobody had gone to the Black Sea, and we knew very little who was there. We knew that these Archaea, Thaumarchaea, they were living in the water column in the suboxic area because they were very happy living there. That we knew. We knew that Anamox bacteria also like to be somewhere here as well. But when we did the first analysis with 16S amplicon sequencing in this water column of the Black Sea, we were totally, our head was blown out by the diversity that was here. And here you have just a little snapshot of how many microbial groups they were encountered and how actually they were changing throughout the water column of the Black Sea to different relative abundances. And of course, apart from looking at the 16S amplicon sequencing to check who was there, we also wanted to know which kind of genomes were there. So we did metagenomics. And here I'm showing you just a snapshot of the different abundances of the genomes that they were found in the water column. And you can see here that there's, for example, Thaumarchaea, this one that I just told you, this Archaea group that really likes to be in the suboxic waters. While, for example, we were very surprised of the huge abundance of certain microbial groups, like Marinomicrobia in the oxinic waters of the Black Sea. And when you look at Archaea specifically, if you blow up this Thaumarchaea genome here, it's right here. And because I'm very much interested in Archaea, you also see that there's an internal diversity of Archaea out there that we were very much interested on, knowing much more what they were doing. And of course, one of the core businesses, of course, is looking at the Archaea lipids. And again, we had the Archaea genome, so we know who was there. We know that all these Archaea were there based on their genomes. And there was a paper in 2018 that we published in Geobiology, in which Martina Solai and Christine Boschman, they actually analyzed the lipid components as well in these samples. And they saw very well that they were the suboxic waters, that they were dominated by Thaumarchaea, this very special Archaea. They were dominated also by Crenarchaea, which is not surprising because we know that it's synthesizing this lipid. And then also in the deep waters, we had other lipids, also membrane spanning lipids that we don't really know who's making them, but now by using lipidomic approaches and by correlating genomics with lipidomics, we know a little bit more of the potential candidates of these lipids. When we look at these lipids, we were wondering about something as well, is that, of course, when I showed you the profile of the genomes in the Black Sea, we saw something quite remarkable, is that when we check what is the expected archaeal lipids based on the archaeal cells we had and accounting for the archaeal diversity we had, taking into account their membrane sizes and so on, we estimated that there was a certain amount of archaeal lipids present in the water column, present there, but actually we detected way more than that as interpolar lipids. And we were wondering if maybe there was an extra producer of archaeal lipids that we missed. That was a big unknown for us. Of course, my core business is to look at Archaea and which kind of lipids they make. But when I went to this water column of the Black Sea and I saw that there were certain groups of bacteria that they were making up to 30% of everybody out there, I also wanted to know which kind of metabolism they had. And I was quite amazed by this purple line over here. And these ones, they were actually members of a bacterial group, an uncultured bacterial group. These are called Cloasimonetes. They are members of the Flavobacteria, Cytophaga, Bacteroidetes superphyla. We detected up to 4 genomes of this group. And of course, they are very interesting because they eat organic matter. So they also are in syntrophy with other organisms. So that's very cool. But what I thought that it was quite funny is that, of course, they are bacteria. And I'm always prone to look at the lipid biosynthetic genes of everything, although I didn't really expect to see something out of the ordinary because these are just bacteria, right? So nevertheless, we were really interested in these Cloasimonetes because they are a very important component of the water column of the Black Sea. I had a PhD student, Sarah Sorminen. She did a fantastic work going out there to the Black Sea and collecting water samples and actually using magnetic beads coated with different substrates, and that we saw that actually these Cloasimonetes, they were very much involved into eating fatty acids and cellulose and dissolved organic matter, and they were very important components in the metabolism of the Black Sea. But that, of course, that was not enough for me because I really wanted to know about their membrane lipids. And I said, okay, I'm going to check. And of course, they are bacteria and they make what they are supposed to make. They have all the fatty acid biosynthesis genes to make fatty acids. They may have the acyl transferases to do the ester bonds. Of course, they have the ability to make glycerol 3-phosphate. So that was fine. But then what it shocked me is that actually they had the ability to make isoprenotes, which is not strange. Many bacteria, they make isoprenotes for making respiratory crinones and pigments. But what I was extremely surprised about seeing is that these bacterial genomes recovered from the Black Sea, they actually did have the prenyl transferases involved into the formation of the ether bonds, as in the archaeal lipid biosynthetic pathway, leading to archaeal membrane lipids. What they did not have, these ones, is that they did not have glycerol 1-phosphate dehydrogenase. We were very surprised about the presence of these 2 prenyl transferases. And when we encounter this, we look at the presence of these ones in other Cloasimonetes genomes recovered from other environments. And we also saw this particular feature in other deep sea environments. Not everybody, not all Cloasimonetes had it. But actually, this feature of having these prenyl transferases was present in all members of the FCB superphyla, which make the hypothesis is, do FCB superphyla members had or have now, in some cases, the ability to make both the bacterial membrane lipids and also the archaeal lipids? Have they have this ability of making a mixed membrane or maybe even a mixed lipid? To be able to confirm it, we still, as we cannot really culture Cloasimonetes, although we have tried many ways of culture Cloasimonetes in the Black Sea, we cannot do it, but we still took the prenyl transferases, the GGP synthase and the DGP synthase of Cloasimonetes, and we put it in an E. coli optimized to produce isoprenoids, like the one of the paper of Caforio in PNAS, and we did lead to the formation also of Archaea lipids in this E. coli. So we did confirm that these genes were functional and that the genes found in this bacterial group Cloasimonetes could actually lead to the formation of Archaea-like lipid, plus the bacterial ones. So we published this in ISME journal in 2020, and we kind of said, okay, well, it kind of seems that we have not such a clear story of the lipid divide anymore. We know that there are organisms out there, or remnants of the microorganisms that they are out there, that they really further give support that at some point during the process of lipid membrane acquisition, either in the last universal common ancestor or maybe even in the transition to a form eukaryotic cells, you can still have a transition with a mixed membrane, and organisms can still have remnants of this genomic feature. So with that, the take-home message I would like to give you today is that when we use lipidomic approaches and metagenomics in the environment, that can be extremely useful. It can be very useful to actually point us in the right direction of which kind of biological sources are actually making membrane spanning lipid of interest, either bacterial or archaeal. Also, when we actually have a microorganism, a model microorganism that we know that it's synthesized the lipid of interest, of course, as you many of you know, it's very important to kind of narrow down which kind of potential candidate, which kind of potential enzymes could be involved into making the steps leading to that final product, so that we can look in the databases of which kind of microorganisms could actually have that capacity. And of course, the use of and the investigation of lipid biosynthetic pathways, either leading to a membrane lipid or any kind of lipid, they can be of very much use to constrain how evolution has been leading to the formation of these lipids. And with this, I would like to finish by thanking all the members of my group. Some of them, they are already gone to very exciting jobs, very good colleagues in my institute that they are collaborators, and all the research vessel and the support staff that help us go from A to B. And of course, the very generous sources of funding that allow us to do what we really like every day. And I would like to thank you for listening. Sorry for the little problems along the way, and I'll be happy to take questions. Thank you.
So, thank you. Oh, muchas gracias. Thank you, Laura. Very interesting.
Muchas gracias.
I thought it was really, really interesting. And when we open, let's say, for questions, I'm not sure if already somebody is.
I'll open the chat as well.
So, well, I'll see. Is there nobody yet? Oh, Otto.
Ah, Otto.
Otto, you want to ask yourself? It's better, huh, than writing.
Puedes pasar al español en cualquier momento, ¿eh?
Hello, Laura.
Hola, ¿qué tal?
Bien, ¿tú? ¿Cómo vas?
Perfecto.
Muy bien. With regard to the sphingolipids in the Black Sea samples, can you correlate them to any bacteria or even to Archaea?
Pues me alegro muchísimo que me hagas esta pregunta, porque estamos en ello. La verdad es que estamos detectando una gran diversidad de sphingolípidos en el Black Sea. Con el network analysis, hemos visto, por ejemplo, una gran cantidad de oxyceramides as well en la redox Klein, y ahora lo que estamos haciendo es intentando correlacionarlo con la presencia al menos de serine palmitoyl transferase, porque no sabemos en ciertos casos qué más genes necesitamos para la formación de estos derivados de sphingolípidos. Nos encantaría saber más. Por supuesto.
Well, So those cyclopentane rings and cyclohexane rings, you correlated to temperature resistance.
Sí.
This would be true also for these other groups of bacteria, the Cetobacteria, the Dermatoga, and the Firmicutes. Is there a correlation? If they're more resistant, they have more of it? Yes.
Yeah. So, well, for the case of the ones of bacteria, and it's not so clear because we don't know. Well, we do see an increase of methylation with an increase of temperature. So there's certain things that we know very well. For the Archaea membrane lipids, for sure, we know that there's an increase of cyclopentane rings with an increase of temperature. Also, pH influences that. At the case of the cyclohexane ring, we don't know what would be the advantage of having the cyclohexane ring. It could be also something taxonomically acquired, and it was useful at some point during the evolutionary process, and it's just there. But we don't have a good explanation about the presence of cyclohexane ring, for example.
Thank you.
I do see some other questions in the chat, so I don't know if you want me to go ahead.
I can read them, or you.
I can go ahead and read it. Don't worry.
But you have to read it loud, because otherwise.
Okay, yeah. Then let's just do it then. Maybe better.
So here's a question from Luis David Alcaraz. I don't know, Luis, you want to ask it yourself in the Zoom room, or should I read it? Because he is here.
Well, maybe I can do that. I don't have a camera right now. Sorry. Great talk. I really enjoy it, and I'm really naive about how hard it could be to correlate the lipidomics profile to the 16S or metagenomes. I was wondering if you make use of, I don't know, maybe standard diversity indexes, or how does that work? I'm totally naive.
Yeah, no, we do diversity indices, but of course, what we do also just like plain correlation, what is high, what is low here, because, and of course, this is really not perfect, because of course, the lipids have a different turnover time than the DNA and so on, so it's really full of problems. But we aim to do this just to kind of narrow down potential candidates. What we do is that not only we have the 16S diversity profiles, but we also have qPCR data so that we can convert the 16S diversity profiles into abundance profile in a way, just by accounting how many copies per genome they would have, etc. And the lipids, we have also certain abundance profiles, of course. So we're just really correlating abundances with abundances.
Thank you.
So we have another question, Laura, in the chat. So this Luis Gerardo Romero Chora from the YouTube channel. He asks, what about the lipids in other seas?
Yeah, so we do these surveys. So we have done this in many other seas. When you go to water columns of, for example, the North Atlantic, like seas that are really turning and that there's a lot of oxygen and so on, the diversity of the lipids is way lower. You do have Archaea lipids as well, because Archaea live in where oxygen is present, but you don't have this amazing diversity that you have in the Black Sea, because in the Black Sea, of course, you have like really so many contrasting conditions that then you have methanes, you have sphingolipids, you have everything, you name it, you have it there, because you have photosynthetic organisms on the top and then anoxic ones. So there's many lipids that are very common of certain environments. And also if you go in the sediments where oxygen is not present, then you start seeing, of course, plasmalogens and any kind of diethers associated with sulfur-reducing bacteria, of course. So that is something that we already know, and we have done it as well.
So let's see. We'll see if there's another question later in the YouTube channel. If nobody else will, I have two questions actually. Laura,
there's another question of Otto.
Otto. Okay, Otto, go ahead.
This is really simple. So what remains is only the glycerol 1-phosphate stereochemistry of the lipid divide.
Yes, yeah, that's still an unbroken rule. We still haven't found an archaeal lipid that it has a glycerol 3-phosphate stereochemistry. But I must say, so I find it difficult to believe that that is something that it could be broken. But it's, of course, very difficult to check the stereochemistry because you need so much biomass to start with and so on. So there's so many organisms out there that we just cannot culture, that we have never really managed to have enough to look at their stereochemistry. So there's also a methodological problem. We cannot really assess that so easily. I can tell you that, for example, all these new Archaea that they are so relevant for the formation of eukaryogenesis, so all the Asgard Archaea, not all of them, they have the glycerol 1-phosphate dehydrogenase. So if they make glycerol 1-phosphate, it's not in the canonical way that all the Archaea make them. It's possible that they make it in another way, but we don't know how they make them. We cannot check them because we cannot culture them in pure culture to be able to get enough biomass to check their stereochemistry. But there's certainly something that is fishy there.
Mm-hmm. Thank you.
Okay, so there's another in the chat from Angelica Mariana Yjara Servin in the YouTube channel. Did you measure the amount of diabolic acid in the superphylum you found at the end? Well,
I'm not really sure if I follow you because they were two different things. So the isodiabolic acid of Thermonarobacter thenolicus, that was from the model organism, the Thermonarobacter. So, and the superphyla, it was of the FCB superphyla. That's what's the Cloasimonetes. It was a different story. So I'm not really sure. Maybe if you can clarify exactly what you really want to ask, because I'm a little bit confused about the question.
So we'll wait for the
question
in the
to
chat.
React.
And I will ask, well, I thought very interesting your approach with the networking of the lipids.
Mm-hmm.
And I wonder how much, if you would make an estimation, how much was known and how much is unknown? I mean, how much could you assign? Like in a percentage, just to guess, like 80% is unknown or more?
Very little, like 5% assign.
You could assign, and the rest is
assign it based on the databases. Yeah.
And did you do this also with other, let's say, type of samples? I mean, these are ocean samples from a specific, but if you do it with soil samples, well, I know you're an ocean institute, but maybe somebody's working.
Yeah. No, we do encounter the same problem. When you go to any environmental sample, the assigned percentage of features is very low. Well, basically all the databases are based on what we have done so far. So not many institutes are contributing to that. And also, of course, databases based on pure cultures of microorganisms or human samples or you name it. So the databases to compare with are very limited.
Okay. And the other doubt I have, so with your Flavobacterium-like organism that has the Archaea type lipids. So what I did get, or let's say, how widespread is this within this phylum? So how many, like, is it only in the ones that grow under high pressure or deep in the ocean, or you also find these genes in, let's say, in this normal Flavobacterium?
Normal Flavobacterium, you can also find them.
So like say the stand organism Flavobacterium johnsoniae, one could look and possibly find.
I think all of them had it.
Okay. Yeah, that's interesting.
The only remarkable feature of Cloasimonetes is that it has both prenyl transferases concatenated, which is not something that you normally find, not even in archaeal genomes. They don't need, because one is cytoplasmatic and the other one is membrane based. So they don't really locate one to each other. So in Cloasimonetes, you have them one next to each other, and that was what we saw very quickly. And it's not that they were horizontally gene transferred in one go, because one of them has more resemblance to certain archaea and the other one to certain other archaea. But all the FCBs, they do have the presence of these two prenyl transferases in some sort of way.
Okay. Well, thank you. I
thanks.
don't know if anybody from our Zoom room wants to ask. Just open your microphone if you want, or give you a few seconds.
I have one other question.
Okay, Otto, go ahead.
So those two C15 units for diabolic acid synthesis, they seem to be connected in the C30 product via the omega-1 Cs. Is this of relevance for the mechanism?
Yeah. Yeah, it's certainly quite relevant. We also have done this for the, so this is for the isodiabolic acids, but Thermotoga also makes diabolic acids, which is C32, and it has a buildup of C16 fused together. And this information has been also relevant to kind of trying to approximate. And in all of these cases, we think, also based on the transcriptome and the proteomic approaches, that they are radical sum proteins that they are making this function. So it's definitely of interest to be able to narrow down the candidates. Yeah.
Mm-hmm. Thank you.
So there's just a
oh
comment.
yeah, exactly. Sorry, I'm not really sure what happened with that slide, so I'm not sure if everybody heard it. So it's good that you got it now. Okay.
So I read it?
Yeah, no, I can't read it. It's fine,
Okay.
don't worry. Yes.
Okay, so there was a doubt with explanation.
Yeah.
So I'm not sure if someone else, any volunteers, more questions? Take your chance. Not every day we have some lipid ecological expert here. Well, anyway, so there don't seem to be any more questions. So thank you very much, Laura. Thanks for taking the time.
Thank you for having me. Muchas gracias por la invitación.
Y estoy seguro que te voy a escribir un correo luego pensando bien, digiriendo la información que platicaste ahorita.
Encantada de que cualquier persona me contacte por el email del principio.
Sí, bueno, pues muchas gracias y... Pues
y buenas noches.
Eso.
Exacto.
Bueno,
chao.
Más tarde.
Muchas gracias.
Hasta luego.
Adiós.
Hasta luego, Laura.
Adiós, Soto.